Next is the differential interference in contrast microscopy. It is also called as Dick microscopy, or we also call it as Nomarsky microscopy or Nomarsky interference contrast microscopy. Dick microscopy, also known as Nomarsky interference contrast microscopy or Nomarsky microscopy, it is used to enhance the contrast in the unstained transparent samples. It is also used for those kind of samples or for those kind of objects which are not easy to stain by the standard methods. Dick works on the principle of the interferometry to gain the information about the optical path length. How we use the Dick microscopy for the working? Dick microscope uses two beams of the light instead of the one. From different ways, the prism split each light beam, adding the contrasting colors to the specimen. And these contrasts are the actual way of the see the objects or the specimens by our own way to the inside the microscope. Image is brightly colored and appears nearly 3 dimensions. When we see the objects from the eyepiece, then we see the image which is seen by the 3 dimensions and which is contrastly colored. What is the principle of Dick microscopy? Light path, unpolarized light enters and becomes the polarized. Then it enters the prism and gets separated into 2 beams. Different kinds of special kind of the prism is used, which is splits the light into 2 different kind of beams. These rays are focused by the condenser. Then there will be the condenser, and the condenser will condense the 2 different beams just like by its own way. And then these rays travel through the specimen, through the objects which is present on this there. Then enter the objective lens, and the second prism recombines the rays, forming an image which is clear through the contrast microscopy. Here is the structure which is given for this different kinds of differential interference and contrast microscopic point of view. From the here you can see from the analyzers, from the point of view you can see the eyepiece which will be here, and the person who is analyzing this, it will be here. And then the Nomarsky prism, the different special kind of prism which is splitting the light into 2 different kinds of beams, which is different. In other microscope, the prism is using which is not splitting it into different kinds of beams. Then there is a condenser, and condenser which again condensed those beams into the one, and then the specimen specimen is clear. And then the specimen which again the 2 different kind of lights which will go to the condenser, and the condenser with the Nomarsky prism, and this is the polarizer. So from here the analyzing objects will be clear from here, and you can see that the contrast microscopy which will make it more clear and more contrasted point of view to see our objects or specimens.